Hi, everybody. My name is Megan Wong. I am in biochemistry and molecular bio in the College of Arts and Sciences. I was advised by Dr. John Caradonna in the Department of Chemistry, and the title of my project is Spectroscopic Analysis of Truncated Tryptophan Hydroxylase Activity. So basically, we're going to be looking at an enzyme called tryptophan hydroxylase, and we're analyzing it using a couple of different techniques that use properties of light in order to figure out how much product we're making with this enzyme. And so to start off, what is tryptophan? So tryptophan is one of the 20 amino acids, which are the building blocks of proteins, and it's shown in the figure below. And hydroxylase is an enzyme that introduces a hydroxyl group, which is an oxygen and a hydrogen, onto a substrate. So in this case, we are using tryptophan, and at that position, we currently have a hydrogen. And with the use of our enzyme, we are adding this oxygen-hydrogen hydroxyl group. And so this is important because tryptophan hydroxylase is part of the aromatic amino acid hydroxylase family, which means that there are a couple other amino acids that go through the same type of reaction using with a similar enzyme. And so first we have phenylalanine hydroxylase, and this converts phenylalanine to tyrosine, and we can see that you still have that same oxygen-hydrogen group added due to this enzyme. And this is important because mutations in this particular enzyme are associated with a condition called phenylketonuria, which is basically when the body can't break down the phenylalanine in the blood and it builds to toxic levels, and it's manifested in humans through brain impairment. And so the other enzyme in this family is tyrosine hydroxylase, and that is used to convert tyrosine to L-DOPA. L-DOPA is the precursor to the neurotransmitter dopamine. And so mutations in this particular enzyme are associated with Parkinson's disease. So when you don't have if you're not able to make L-DOPA, you're not able to make dopamine, and you get that, you can see that with Parkinson's. And so the last enzyme of this family is tryptophan hydroxylase, which is the one that we're looking at. And so from here on out, we're basically only going to be looking at this bottom reaction and this enzyme. So TrpH, that's the enzyme that we're looking at, and then the product is on the right and the starting material is on the left. So basically we're going to be measuring the amount of the right molecule product that we're forming over time. And so why tryptophan hydroxylase specifically? So the product from the enzyme's reaction is used to make serotonin in the midbrain. And so this is important because we know that deficits in serotonin are associated with conditions like depression, insomnia, and anxiety. And so if we're able to figure out how this enzyme is regulated and how it functions in the body, then we can better understand some of these conditions. And so our hypothesis was that if we took a form of this enzyme that removed part of what we thought would be the regulatory domain and would cause it to be active within the body, then we would get higher activity compared to the reported values that we're seeing in brain tissues. And this is because in tissues, we might have a combination of active and inactive protein. And because of this, then we might be seeing lower activity levels than when we see with purified enzyme with the other ones of the family. And so to look at this, we use protein expression and purification, and then we use two different types of spectroscopy, which use the property of the product to emit light with the fluorescence spectroscopy, and then with the property of the product to absorb different wavelengths of light with the UV-Vis analysis. And so our first goal was to express and purify this specific enzyme. And so basically what's going on in this figure is this is a gel. It separates all of the pieces in our sample by size. And so the first lane is used as a standard, and that's just to show us where we would expect to see molecules of whichever size. And then the next 4 lanes are used to show that we were basically washing off all of the extra products that were made during the expression process of our enzyme. So then finally in the last row where the red box is, that's where our enzyme is. So we have this strong expression, which is seen by the darker band, and we're able to confirm that we made the correct enzyme because of where it lies in relation to the standards. So it's at the right size that we know that it's at, and we have a lot of it to use in our future experiments. So our second goal was to look at fluorescence spectroscopy, which basically uses the property of the product to emit at a certain wavelength of light. And so we're measuring the intensity of fluorescence over time. So we measured it for 60 seconds, and basically we put everything in with our enzyme, and we were making product. And as we made product, the intensity increased because the product had a greater fluorescence intensity compared to the starting material. And so we were able to track that and calculate a rate based on the first 20 seconds of the measurement. And so that's where the orange line is. We basically took a linear trend line of that and found the slope, and that was our rate for that experiment. So we ran this at a lot of different experiments, changing the amount of starting material that we have. And so what we would expect that would be that as we increase the amount of starting material, which is measured on the x-axis, as we increase that, we would expect that the overall rate of product formation would be faster until we reach a saturation point. So at some point, the enzyme is only making as much product as it can, and it would just kind of level off at the top. So we started to see that we do get an increase at the lower concentrations, and then it eventually started to show some sort of inhibition, which was not what we expected. So we went to look at just the starting material, and we saw the same thing with the starting material. We thought it would increase as we added more of the starting material to the sample. However, we get some sort of inhibition. So from this, we concluded that the fluorescence spectroscopy was not the correct method for analyzing this enzyme. And we turned to UV-Vis, which uses the property of the product to absorb light. And we determined that 315 nanometers, other things in our reaction are not changing in absorbance. And so we wanted to use that wavelength so that when we look at our product specifically, we know that any changes in absorbance are due to the product formation and not because of other things in the sample that are changing in absorbance over time. And then we confirmed this by looking at our starting material and our product together, and we saw that at 315 nanometers, we do get a distinguishable difference between the absorbance between our starting material and product, which means that we should be able to track this change in absorbance over time as we did with fluorescence, but hopefully to overcome those challenges that we had with concentration over time. So in conclusion, we were successfully able to purify the protein. We determined that fluorescence spectroscopy was not the appropriate method for analyzing this enzyme's activity, and we determined a need to turn to a different form of analyzing this enzyme to be able to fill the gaps that fluorescence could not. I'd just like to thank KHC, my advisor, John Caradonna, and the other two members of our lab, Cynthia and Bailey. Thank you.
Really great job. That was awesome. You mentioned that there was other work that would need to be done to fill in those gaps. Do you have any idea of what kind of work going forward would look like for filling in those gaps?
Yeah, so we're still in the process of optimizing the same type of experiment, but with the UV-Vis spectroscopy. So the first couple of slides with that was basically just like making sure that we know what wavelength we want to use. But then from that, we're basically adapting what we use for fluorescence and then using that and just kind of using that wavelength that we just determined, running the same experiments, but just measuring the absorbance instead of the fluorescence. And hopefully we won't see any issues because we know that the tryptophan with concentration, at least it's linear within the UV-Vis region instead of looking at the fluorescence, which did not turn out to be linear.
So I know you mentioned that the fluorescence didn't work, right? But is that in itself a discovery, this inhibition at these concentrations and so forth, or was that maybe known about the enzyme in a different way?
Yeah, so actually what was interesting about the fluorescence was that we took a procedure that was previously published, and they were showing that we were getting like what we would expect. So we were thinking, okay, if we just replicate this, then we will be able to see what we expect and that everything will turn out fine, and we'll be able to like talk about the activity and stuff like that, which like we didn't quite get to do with this because the fluorescence didn't work out. But because we modified the way that they did their procedure, they were adding in more iron into the cofactor like region. And so we basically like took that step out and just had our active enzyme, and then from there we were hoping to see that, so we didn't. So like it is still moving that forward. So it kind of more, I guess, turned us to the UV-Vis, but kind of just away from fluorescence, which was the main focus originally.
Okay, and then just one really technical question,
Yeah.
because I'm curious. So you had some nice isospecific behavior in your
Yes.
UV-Vis data. What is the change there that's taking place?
Yeah, so we took the cofactor, the pterin cofactor, and we just measured it over time, so each line is at a different minute, and we know that as that oxidizes, it changes in absorbance. And so we actually see 2 isospecific points. So isospecific points is like where we're not seeing a change in absorbance, so where the lines are all intersecting. So around 275, we also have one, and then we have the second one at 315. But the reason that we looked at the 315 was because if you see at 275 nanometers, we actually don't get a difference between the product and our starting material, so that wouldn't be a good wavelength for us to use in our experiment.
Any other questions for Megan? All right, if not, please join me in thanking Megan again for a nice talk.
Thank you.